Okay, so welcome to Biochemistry 501, Lecture 3. Today's topic, we're going to be talking about DNA replication, that is the process of actually taking the genome, the genetic material of the cell, and making a complete and faithful copy of that material so that when the cell divides, each daughter cell will get a copy of this genetic material. And for the purpose of this lecture, we're going to learn about the chemical mechanisms and enzymes that are involved in actually performing that synthesis, as well as the other additional enzymatic activities that will be required to help facilitate this copying process, because it's going to turn out that simply being able to synthesize nucleotides is not going to be enough to deal with all the challenges that come when you try to actually replicate the entire genome. So as with all the other lectures, I'm putting the learning objectives here that you should pay attention to during the lecture to make sure you follow. And kind of as an overarching outline of this, for these lectures, we're going to learn about replication kind of from three different vantage points. We're going to learn about the chemical mechanism of DNA synthesis, so where the electrons are going and which atoms are interacting with which to drive synthesis. And then we're going to learn about DNA polymerases, which are the enzymes that are responsible for actually catalyzing the DNA synthesis. We're going to learn about how those work and how they even have proofreading capabilities that help them perform this synthesis process more accurately. And then finally, we're going to learn about how that polymerase activity is embedded in a larger context with other helper activities to actually be able to copy the genome, because there's a lot of things that are going to have to happen to copy the genome. You're going to need to unwind the DNA, and there's some topological constraints that come on. And so we'll learn about how those other components contribute, or how these other enzymatic activities help to ensure we can actually replicate the genome. So let's get started just thinking about the chemical process of replication. And when we're talking about genome replication, what we're really talking about is using a template strand in our genome to provide the complementary nucleotide strands for the other synthesis. And it turns out that the process of genome replication is called semi-conservative. And what that means is that after you copy the genetic material from the parent into two copies, so if we started with one parent helix and we copied it into two daughter helices, semi-conservative means that each daughter helix is actually going to get one of those strands completely from the parent, and then the other complementary strand will be synthesized completely new. And so in this semi-conservative, you should be thinking that the parent strand of DNA is going to serve as a template to generate the daughter strand. And so now let's think about what this means in terms of using a template strand to add complementary nucleotides. So imagine that we had our synthesis strand here growing, like you see here, and the template strand is over on this side. And remember, 5' to 3', 5' to 3' here. And so what we're asking the cell to do is to recognize that because the template strand has a C in this position, we should specifically add a G to this location. So that's what needs to happen, right? And so how does this happen at a chemical level? Well, we actually talked in the very first lecture about the chemical mechanisms of nucleotide addition, in which at the 3' end of our strand that's undergoing synthesis, a nucleophilic base is going to, a base is going to activate this hydroxyl group here to facilitate a nucleophilic attack. And what it's going to perform this nucleophilic attack on is a nucleoside triphosphate, in which the pyrophosphate here can function as a nice leaving group to drive the synthesis. And so you've seen this chemical mechanism before, but I'm just showing it to you again so that we can make sure we really understand the chemistry that's going on to make these phosphodiester linkages. Because it's going to turn out that this is the chemistry that the enzyme polymerase is going to have to perform for us. And so just to put it in perspective here, again, if this is the process that we're trying to perform, a base is going to activate this 3' hydroxyl to facilitate nucleophilic attack. And in that way, we're going to add a new nucleotide in our synthesis strand. Now, as I said before, that's the chemistry, but that chemistry is not just going to happen very effectively if you just mix those reagents together. And like everything you've learned in the class, there's going to be an enzyme that's going to serve as a catalyst to facilitate and accelerate this reaction to go forward. And the enzymes that perform this catalyzed nucleotide addition in cells are called DNA polymerases. And so sort of abstractly, you can think of them as these objects that are going to interact with your template strand and the synthesis strand, and they're going to add the DNTPs to the end of the strand here. And some key facts that you need to know about DNA polymerase is that the substrates for DNA polymerase are DNTPs, which are the triphosphate, they're nucleoside triphosphates, and that's because the additional two phosphates make for an excellent leaving group to drive the polymerization reaction forward. They don't just add DNTPs arbitrarily, they're only going to add them in a manner which is complementary to the template DNA strand, and that's of course going to be important because our goal here is to make a synthesized strand that is complementary to a template. So those DNTPs need to be base pairing with the appropriate end of the template strand. An important topological thing to know is that only the 3' end of the substrate can have the DNTPs added to it. So you're not going to get polymerase adding DNTPs to the 5' phosphate. It's only this 3' OH which can perform the nucleophilic attack on a nucleotide. That's where the action is going to be. And then finally, a really important thing to know as well is that these enzymes can only perform synthesis. The synthesis can only proceed off of an already existing double-stranded DNA fragment. That is, if you look at this picture here, the polymerase is going to be happy to add a nucleotide because there's already this stretch of double-stranded DNA that's formed between the template and the synthesis strand. But if this template isn't here already, that is, if there's nothing to extend off of, then polymerase is actually not going to be able to perform this reaction very well. And so we say that polymerase requires a short primer, which is what we call this short piece of DNA that anneals to our template strand in order to initiate synthesis. And that's going to be really important for thinking about the overall process of genome replication, as you'll see. Now, what is it that polymerase actually does here to make this chemistry happen? So we have this beautiful structure here of a DNA polymerase with the template strand here in pink entering the active site and our newly synthesized strand in orange. As you can see, this is that sort of double-stranded stretch between template and synthesized strand that I mentioned before. And the new nucleotide that we're going to add via base pairing is going to happen right here. And so from a structural point of view, people often describe the polymerase as looking sort of like a, looking sort of like a claw or a hand, in which the DNA sort of rests on the palm in between the thumb and fingers of this structure. I just show it to you here because it's sort of a beautiful enzyme, but also it really highlights that you can see that this existing stretch of template and synthesized strand forming this double helix is going to make it very natural to place the correct nucleotide in this position. Now, beyond just the structure of that polymerase, remember that the polymerase needs to actually catalyze that nucleotide addition reaction we showed you before. And so abstractly, we know that a base needs to come along and activate this hydroxyl, and this hydroxyl will perform nucleophilic attack on our DNTP to form the phosphodiester. And so as an enzyme, what is this polymerase actually doing to facilitate this reaction? Well, it turns out that the polymerase makes substantial use of magnesium ions, so basically using metals to help drive this reaction forward. And so polymerase contains these aspartate residues, which can help coordinate these magnesium ions that you see here. And those magnesium ions carry positive charge, and that positive charge helps activate this process through two mechanisms. So one, the positive charges can help facilitate the activation of your 3' hydroxyl here to make it more nucleophilic. So this is going to help with making the 3' hydroxyl primed to attack our DNTP. And then the other thing that the polymerase can do that's really helpful with these metal ions is those DNTP nucleotides carry a lot of negative charge, right? So they've got 3 phosphates on there. There's a lot of negative charge. These magnesium ions can help stabilize that negative charge that is present on these molecules. And the role of the metals is so important that if you take metals out of a chemical reaction using polymerase, so if you were in the lab and you were trying to perform a reaction using these enzymes, if you don't have these metal ions in solution, then no chemistry is going to happen. So in a certain sense, this polymerase is an enormous object who one of its primary functions is to grab these metals and put them in the right place to make this reaction go. Now, an interesting thing to ask yourself is, well, if we look back here, you know, the chemistry looks solid, like we can make, we can connect these things together. Why do I actually need a primer in order for this reaction to go? Like, why can't I just add, why couldn't I just mix two nucleotides together and just have polymerase connect them nicely? And so some of the answer to this comes from the fact that you'll recall that in lecture number 1, we learned that in the context of a double-stranded DNA B-form helix, base pairing provides only a small portion of the energy that stabilizes the structure. So we get these hydrogen bonds between A and T, for example, but it's the base stacking interactions of those nucleotides with other Watson-Crick-Franklin base pairs that provides important steric and electronic interactions that help stabilize and reinforce the base pairing of that whole B-form DNA structure. Therefore, these base stacking interactions, or sorry, these base pairing interactions between A and T are going to end up being much more robust and more specific in the context of an existing double-stranded DNA structure. So if you already have this part stacked, then forming a proper base pairing interaction at the growing end is going to be much more favorable than if you just tried to make that happen on its own. So that's one of the reasons why the primer, a primer in particular, that double-stranded stretch of DNA is really important to be able to drive this reaction forward. Okay, so to sum up what we've learned about DNA synthesis by DNA polymerase, we've told you about the chemical mechanism. So we're going to have that same base activation of hydroxyl for a nucleophilic attack on the 5' phosphate of a DNTP. Thus, synthesis is always going to proceed in the 5' to 3' directions. We're never going to add nucleotides to the 5' end of a growing strand. It's always going to be the 3' end. That's super important to make sure you know. Polymerases work by using metals that they bind to facilitate the nucleophilic attack and stabilize phosphate charges. And very importantly, DNA polymerase requires a short stretch of existing double-stranded DNA to synthesize off of, and we call that short piece of DNA that is annealed to our template the primer. And the double-stranded DNA stretch or primer facilitates the addition of complementary base pairs by taking advantage of the additional energy that comes from base stacking. So hydrogen bonding is between complementary base pairs is what we're looking to achieve, and the stacking interactions help ensure that we select for that particular type of hydrogen bonding pattern that you see. Now, this process in the cell is actually very fast and very accurate, but mistakes will happen. That is, the polymerase will not always add the correct nucleotide. And so what happens if DNA polymerase makes a mistake? Obviously, it's going to be a huge problem if we start messing up the contents of our genomes, because those are where the genes live. They're what encode protein sequences. So if we start making, if we start putting errors in our genome, we're going to lose the fidelity of that information. And so in the next video, we're going to find out how DNA polymerase actually deals with mistakes and how it has proofreading activities that let it go back and fix mistakes that it makes along the way. And so I'll see you in that video.